The protein, known as NusG, pauses the transcription machinery at specific DNA sequences to facilitate what is called intrinsic termination, and prevent unwanted transcription that could disrupt cellular function. A new study, led by Penn State researchers, shows that NusG and the related protein, NusA, together facilitate termination at about 88% of the intrinsic terminators in the bacteria Bacillus subtilis. Understanding this process expands our basic knowledge of this key cellular function and could eventually aid in the development of antibiotics that target and disrupt gene regulation in bacteria. A paper describing the research appears online in the journal eLife. For a cell to access the genetic information stored in DNA, it first must be transcribed into RNA by an enzyme called RNA polymerase, said Zachary F. Mandell, a graduate student at Penn State and first author of the paper. This process is highly coordinated to ensure that the right genes are expressed at the right times and at appropriate levels for the cell to function properly. We are interested in understanding the mechanisms that allow the cell to stop transcription at precise locations along the genome. Proper regulation of gene expression occurs in three basic stages. First, transcription is initiated by RNA polymerase that binds to the DNA at the beginning of the sequence that is being transcribed. NusA and NusG then bind to RNA polymerase during the elongation process to make an RNA copy of the DNA sequence. Finally, the transcription must be terminated at the appropriate spots in the genome. Termination of transcription is especially important in bacteria because the genes are packed tightly together along the genome, such that failure to terminate transcription at the right locations could lead to inappropriate gene expression, said Paul Babitzke, professor of biochemistry and Mole. The mechanisms for termination in bacteria traditionally have been classified as either factor-dependent, which relies on a protein called Rho, or intrinsic termination, which was thought to be factor-independent. Intrinsic termination relies on an RNA hairpin structure that forms in the RNA. NusG and NusA are both proteins classified as transcription factor that help regulate gene expression and are part of the complex of proteins that read and transcribe DNA during elongation. NusA interacts with the RNA molecule being produced and NusG can bind to the DNA to pause elongation. Based on previous research, NusA is thought to play a role in intrinsic termination by aiding in the formation of the RNA hairpin, but the role of NusG in termination had not been established. To further explore the role of these two proteins in intrinsic termination, the research team produced strains of bacteria that lacked NusA, lacked NusG, and lacked both NusA and NusG. Thanks for watching. Please subscribe my channel for more updates.